Good morning, everyone. I'm Anusha A. I'm a DST CSRI postdoctoral fellow at the Medical Intelligence and Language Engineering Lab, headed by Professor Dr. A.G. Ramakrishnan of the Department of Electrical Engineering at the Indian Institute of Science. One of the focus areas of research at our lab is the exploration of the functional connectivity of the human brain during slow, deep breathing, during sleep, consciousness, and its various alterations. This work, entitled Differential Effects of Slow Deep Inhalation and Exhalation on Brain Functional Connectivity, was carried out in our lab by Mr. Suvam Dey, who interned in our lab last summer as a part of the Indian Academy of Sciences Summer Research Fellowship Program. Breathing, as we all know, is a fundamental process of the human body. It is mostly an involuntary and rhythmic act that continues during sleep and even when a person is unconscious. However, we also know breathing as a voluntary act because we know that a person can control breathing during talking, singing, or even while doing some voluntary breathing exercises. Several years of research have led to the understanding that these involuntary and voluntary breath control systems emerge from separate sites in the central nervous system and have separate descending pathways. Involuntary control involves neuronal assemblies in the hindbrain, specifically the pons and the medulla breathing control centers in the brainstem, and the voluntary control of breathing emerges from the motor and the premotor cortex of the brain and stimulates the neurons that innervates the diaphragm and the intercostal muscles. The schematic on the right here represents the neurophysiological techniques that are used to assess respiratory muscle control. So the multiple dissenting pathways of breath control that we already mentioned integrate at the respiratory motor neurons and determine the functional output of the muscles. So using electromyography or EMG, the output can be measured during resting breathing, exercise, or as evoked signals in response to transcranial magnetic stimulation or TMS over the motor areas or the phrenic nerve stimulation, that is PNS, of the peripheral nerve. The output from cortical networks can be measured using electroencephalography or EEG. So this study attempts to understand functional variations in the cortical regions of the human brain during slow breathing using EEG. The inhalation and exhalation phases of the breathing cycle are analyzed to see if the brain rewires itself during these phases to assign distinct functional connectivity signatures for them. Functional connectivity is one of the three modes of brain connectivity. Brain connectivity is operationally defined as the estimation of relation between distinct units within a nervous system. These units can be individual neurons, neuronal assemblies, or anatomically segregated brain regions like the right and left hemispheres of the brain or the anterior and posterior regions of the brain, etc. So brain connectivity is broadly classified into structural connectivity, functional connectivity, and effective connectivity. While structural connectivity refers to the anatomical connections linking the different neuronal assemblies, functional connectivity is fundamentally a statistical concept. So in very generic terms, functional connectivity captures the statistical relationships between the distributed and spatially remote neuronal units. It is often calculated between all elements of a system, regardless of whether these elements are anatomically connected or not. So it should be kept in mind that the functional connectivity does not make any explicit reference to specific directional effects or an underlying structural model. The third mode is the effective connectivity, and it may be viewed as the union of structural and functional connectivity. It usually describes the directional effects of one neural assembly over another. So basically, one neuronal assembly or region in the brain causes a perturbation in some other neuronal assembly or region, and effective connectivity captures such perturbations along with the directions. So in this study, we focus on the functional connectivity between different regions of the brain, and the regions here is being defined by the scalp EEG electrode locations. Moving on to the study objectives, while most of the existing literature on the neurophysiology of breathing has focused on autonomic brainstem processes, the higher brain mechanisms underlying the cognitive aspects of breathing have not been explored much. Recent years have seen an increasing interest in understanding breathing as a complex behavior involving cortical areas above the brainstem. This study is envisaged as a precursor to understanding functional variations in the human brain during breathing using EEG. The inhalation and exhalation phases of breathing cycle at 2 breaths per minute are analyzed to see if the brain rewires itself during these phases. We also take an exploratory look at the intercortical and the intracortical distribution of functional connections in the study. We believe that the study will aid similar investigations that attempt to explore how breathing rhythms modulate brain scale functional connections. So in order to meet these objectives, we devised a breath study protocol. We administered a slow breathing protocol at 2 CPM on a total of 20 subjects, 11 males and 9 females, within an age range of 23 to 60 years and an interquartile range of 10.3 years. All these subjects who volunteered to participate in the study were right-handed and in self-reported good health with no signs or history of any acute or chronic respiratory pathologies. Of course, a written informed consent was obtained from all participants. The study protocol extended over an approximate duration of 15 minutes, and during the entire duration, the subject sits upright and relaxed. The protocol involved baseline and paced breathing sessions. Paced breathing was performed at a breathing rate of 2 cycles per minute for a duration of 2 minutes. A visual metronome was designed and developed in-house to help subjects synchronize their breathing during the paced breathing sessions. The metronome has 2 concentric circles as shown here. The inner circle slowly grows in diameter from the center to reach the boundary of the outer circle to depict the inhale phase. Similarly, the inner circle contracts from the boundary of the outer circle to the center to depict the exhale phase. The participants fix their gaze at the center and synchronize breathing to the metronome. EEG data during baseline and paced breathing sessions were recorded from all subjects using the Ego MyLab system from A&T Neuro. The system constitutes an Ego amplifier and a 64-channel waveguard EEG cap with AG-AGCL electrodes embedded in the standard 10-10 layout. And the whole brain was topographically divided into 4 regions as shown in the layout diagram here. We considered the frontal, temporal, central, and parieto-occipital regions to explore the intercortical and the intracortical distribution of functional connections. EEG data was recorded at a sampling frequency of 1 kilohertz. So what you see here is an illustrative photograph of the breath data collection protocol being administered to a male subject. The subject, as can be seen, is following a visual cue while breathing. He's wearing an EEG cap. ECG electrodes can be seen on the wrist, GSR electrodes on the fingers, and a chest belt that measures the respiratory effort is also worn by the subject. And the visual metronome displayed on the screen may be seen on the right side photograph. Now, data collection was followed by pre-processing. All pre-processing of EEG data was done using EEG Lab version 13.4. EEG data collected from each participant during slow breathing phases at 2 cycles per minute were imported into EEG Lab and subjected to line noise removal using the CleanLine EEG Lab plugin. This plugin was used to adaptively estimate and remove line noise from EEG channels using the multi-tapering and Thomson F statistics. Channels containing excessive artifacts were removed from the data to be interpolated later on in the pre-processing pipeline. EEG epochs of 2.5 seconds were generated from inhale and exhale phases, and a total of 603 epochs were generated. Further, these epochs were visually inspected to discard those containing artifacts due to jaw clenching or head or electrode cable movement. And a piecewise detrending on the data was done to remove any trends, and subsequently independent component analysis was done on the data. After removing the artifact-specific independent components selected by the IC algorithm, data was reconstructed for each phase. And also at this stage, the data from previously removed channels were interpolated using a spherical spline interpolation method, and the clean EEG data thus generated were used for further analysis. Now, the functional connectivity between two EEG signals was computed using the phase slope index, which is a frequency domain bivariate FC measure. It estimates the consistency of the phase lag or lead between two signals as a function of frequency. Specifically, if there is a constant time lag between two signals, as in if a signal from one structure consistently leads the other, then there is a linear relationship between phase lag and frequency, and the slope of that line indicates the sign and magnitude of the time lag. We chose to use the PSI measure here because PSI is relatively insensitive to false positives. And also in the example shown here, we'll see that the red signal always leads the blue signal by 5 milliseconds, which result in a different phase lag across the frequencies 20 hertz, 25 hertz, and 33.3 hertz. So this would result in a positive slope for the red-blue phase difference, which is indicating a red lead in the slope graph given below. So a PSI value that is deviating substantially from zero for a wider frequency range suggests that one of the signal is consistently leading the other, thereby indicating an interaction between them. Now, the identification of brain functional networks that could efficiently distinguish the inhale and exhale phases of slow respiratory cycle was formulated as a binary classification problem with inhale and exhale as the target classes and functional connections as features. PSI was estimated for 7 frequency bands for all possible pairs of EEG time series data recorded from 61 scalp electrode locations. So that would constitute a vector of 1,830 functional connections for each epoch. Now, t-test based feature selection was performed to identify the most optimal and relevant subset of functional connections that could effectively distinguish the target classes. Features that would yield statistically significant differences between inhale and exhale classes with a significant threshold of 0.05 were identified, and these were fed as input to the classifier. For machine learning algorithms, we attempted a linear discriminant analysis, quadratic discriminant analysis, support vector machines with both linear kernel and the radial basis function. And these 4 algorithms were tested for their utility to distinguish inhale from the exhale phase. A 10-fold cross-validation accuracy was used as the performance metric. Feature selection and classification both were performed separately for each of the EEG bands mentioned here. Moving on to results, the performance of different classifiers at distinguishing inhale and exhale phases during different EEG bands is shown in this table. So as can be seen, the SVM RBF yielded the highest cross-validation accuracy of 99.08% in the low gamma band while utilizing 340 functional connections. It may also be noted that the performance metrics for SPM with RBF kernel were higher than the linear kernel, which may be attributed to its better generalization power and the formation of a more robust model. Results also highlight that slow inhalation and exhalation can modulate high-frequency oscillations such as gamma activity, which are known to govern higher cognitive processes like problem-solving, decision-making, and language skills. The figure on the left summarizes the intercortical and the intracortical distribution of 340 low gamma band functional connections that yielded the highest classification accuracy. So, as can be seen, 230 of the total 340 connections were intracortical, with the majority of them between the central and the parietal regions of the brain. The intercortical connections were maximum at the central region. And this is also in agreement with the existing literature that mapped cortical origins of the respiratory-related evoked potentials to the centroparietal cortex of the brain. The strength of these inter- and intracortical functional connections during inhale and exhale phases were further compared statistically using the Wilcoxon sign rank test to tease out any significant variations. And the results are summarized in the figure on the right. As can be seen, the Gamma oscillations show higher phase synchronization, which is indicated by the higher average PSI during slow exhalation in all regions, except the frontal region of the brain. Also, the increase in phase synchronization during also this increase in phase also this variation in phase synchronization is statistically significant for the CF, FT, CT, CC, CP, PT, and TT connections, thus suggesting respiratory phase-related entrainment in the brain. So taken together, the results are in agreement with the hypothesis that inhalation and exhalation phases of slow, deep breathing have a distinct influence on the functional connectivity within and between different cortical regions of the human brain. This study sheds light on the inner and concluding, this study sheds light on the inter and intracortical functional connectivity variations in the brain during conscious, slow, and symmetric breathing. Results suggest that a low gamma functional connectome, which is known to govern higher cognitive processes, can distinguish between the inspiration and expiration phases accurately. It is further observed that a majority of these connections were within central or between central and parieto-occipital regions. These findings may help understand the complex neural mechanisms involved in consciously controlled breathing and possibly explain the therapeutic effects of practices like pranayama or meditation, which often use slow breathing techniques. Thank you.